now we know exists as the trimeric form. And then this particular protein are very important for viral infection and also a major target for antibody response. Over the years, my group at Tsinghua University, together with Professor Xinchen Wang, has been studying the structure-function relationship between the coronavirus spike protein and antibody response. This graph highlights the current stage of knowledge, of our knowledge in the field, how the conformational structure changes during the course of the infection. As you can see, the trimeric structure protein spike doesn't state as a standstill conformation, rather a constant change in shape and in position. In particular, RBD involving the so-called up and down conformation that's defined as either an inactive pre-fusion state or active pre-fusion state. For viral infection, the RBD have to be in the up conformation in order to bind ACE2, which trigger a cascade events that leading to the ultimate fusion between viral and the cell membrane. So this conformational structure really trigger a lot of academic interest in the field and also pose some challenges for our human immune system to recognize this conformational change. So our group and Dr. Wang's group are among the leading groups of publishing those dynamic changes for SARS coronaviruses and for MERS coronaviruses years before COVID-19 outbreak happened. Since we have a very good scientific tools in the lab, so when the sequence of COVID-19 or SARS-CoV-2 released in the database, my group and Dr. Wang has immediately synthesized the spike protein as well as RBD protein in the lab and purified it in cold crystal with ACE2. So our group, again, among the first few groups that published the crystal structure of ACE2 and RBD, which highlighted in the middle. The green is ACE2, and then the cyan color is representing the RBD. The red represents the receptor binding motif that actually interact at the interface between ACE2 and RBD. The first revealing of this structure indicates this structure has a dramatic similarity with the SARS-RBD and ACE2 binding, suggesting both viruses share a very conformational similarity. At the beginning, we thought that since these have a tremendous degree of similarity in structure, they may have a very high level of recognition, in particular antibody cross-reactivity. But it turned out to be much less than we initially expected because of the red region highlighted here are much more divergent than the structural biology show. It's revealed about only 50% in sequence. So that really triggered a lot of downstream study to study not just the specific antibody responses against COVID-19 or SARS-2, but also the cross-reactivity between SARS-1 and the MERS. If you put this structure onto the trimeric format, you could see the ACE2 is only able to bind to the RBD in the standing up form. So this is very consistent with the later on study that showed or confirmed by many other groups in the world, in the field. Once we resolve those structure, we have a very important protein, including RBD and trimeric protein, as a bait, in particular in functional relevant state, which allow us to use them as a bait to fish out the B cells that really producing the COVID-19 or SARS-CoV-2 antibodies. As we all know well that when humans are infected by any viral or bacterial or fungal pathogens, that we produce tremendous amount of antibodies. Those antibodies largely targeted to the surface protein and also some of the structural protein, not necessarily on the surface. Among those antibodies produced, only a few good antibodies that are able to block viral infection. So we term those antibodies as a neutralizing antibodies or potent neutralizing antibodies. So isolating those and characterizing those neutralizing antibodies become one of the major focuses in the field of infection and immunology. And studying those antibodies are very important because that not only can use those antibodies to understand the antibody and pathogen interactions, pathogenesis, and also perhaps use those antibodies to further develop as antibody drugs, but also provide a very important and precise target for our future vaccine development. On the picture on the right really highlights the crystal structure of antibodies in its full form. As you can see, antibody exists as a dimeric form, and each monomer consisting of a heavy chain here labeled in blue or yellow, and a light chain here labeled in red. In our plasma, we have about 20% of plasma protein made of this antibody, suggesting antibody play a very important role in protecting from diseases and also attenuate disease progression. Over the years, in my laboratories, we have been constructing, automizing, and also doing the B-cell and antibody studies for many infectious diseases, in particular here in China, including HIV, influenza, and MERS, and the Zika, and many other, in particular, viral pathogens. So the general theme is quite straightforward, but over the years, there's a lot of sophisticated technologies has been plugged into the process, facilitating the rapid and much more high throughput screening of B cells, antigen-specific B cells from the infected individual, allowing us to carry out much more comprehensive antibody studies than before. So the process starts with collecting the patients, either is infected or most likely the convalescent individuals. From those individuals, we collect a few ml of blood. In most cases, about 5 to 10 mLs. With those blood in hand, we could do a lot of study like the Scott Boyd has just presented. We could use plasma to do all this sequential IgD, IgM, IgA, IgG studies, and also the subtyping studies associated with binding, neutralizing analysis, and then the antibody profilings. But the first and foremost, we use those antibodies, we use this blood to do is to really isolate those antigen-specific B cells that perhaps may yield some good neutralizing antibodies. The graph on the top right really highlights the B cell that really recognize RBD or trimeric SARS-CoV-2. And then we sorting those cells into the single wells. And then we could do two things. Either we amplify antibody heavy light chains from the single B cells, or we do a single cell sequencing to capture the whole transcriptome of profiling of that particular B cells. Once we capture the heavy and light chain sequences, and we could cloning them or we could synthesize them and put them back into cell culture flags to produce those antibodies individually, and then characterize them for binding, neutralizing activity among those with a good neutralizing activity, and then we produce and manufacture for the ultimate human test for either use for prophylactic purposes or therapeutic purposes. So we use this procedure, have successfully isolated among the first group of in the world, isolated Zika human neutralizing antibodies, and MERS human neutralizing antibody, and some of the influenza, and demonstrated tremendous of neutralizing activity, but also protectivity in animal model. So since we have this technology, and when the COVID-19 outbreak occurred in China, we immediately enrolled the patient. By that time, we don't have the luxury of grouping the patient into just a Scott show. They have a few hundreds. So whoever we could get, we just get the blood and put them into our starting cohort and characterize them as much as we could. The first cohort we studied, in fact, include 8 individuals. Here are highlighted in different color, but the red color are the ones that demonstrated severe stage of infections. They all need to be hospitalized. They have in ICUs, while the other 5 highlighted in blue are reasonably mild patients. I need to mention that the first 5 patients, representing perhaps the most important medical-related patients in the COVID-19 epidemic. These 5 patients were first documented in Shenzhen, Hong Kong University Hospital. Those are the first cluster with a solid evidence suggesting the SARS-CoV-2 is a human transmissible or airborne transmission disease. Really highlights and give a first scientific warning to the entire field that this disease is airborne disease. So they all had the family group had a travel history to Wuhan. When they came back to Shenzhen, brought them back to Shenzhen, and then they transmitted within their family cluster. So when they arrive in Shenzhen, and we collaborate with them and get the blood sample, you could see many of the blood samples highlighted here were collected during the 2nd or the 3rd half of January. While their symptom onset occurred during the early of January, like January 3rd, January 2nd, and January 18th. So you could see this really marked the early cluster of patients that identified here in Shenzhen, perhaps very early in China as well. And also interesting point, we were lucky in a way that although we only have 8 patients, but those patients have mild diseases, some are very severe diseases. And you can see those severe ones are all about 65-year-old, suggesting the age relatedness with disease status is quite clear. Although many things that we are not certain, still not certain. I saw Dr. Boyd present a beautiful data about this disease severity and antibody responses, but we are still not in a position to provide a concrete causative relationship between our immune responses and the disease status. So from those 8 individuals, we using the single cell sorting technology, and we plug into the RBD, in particular for the first batch of study, we plug into the RBD labeled protein. We have isolated over several hundreds of SARS-CoV-2 specific BCR sequences. Here I just show 3 individuals. On the A column, you can see the percentage of RBD specific B cells varies from individual to individual. Some are really at the low level, like 0.13%. Some in the bottom patient is like 1.86%, so it's about 10-fold differences. In terms of genetic diversity, for heavy chain in the middle column and the light chain on the right column, you can see their distributions and clonal expansion that varies from person to person. Some has for the first, you can see there's 3 clones are really expanded in their antibody repertoire for both heavy and light chains. And then for second individual, there's one particular family expanded so dramatically, 33%. And then of course, that may be skewed because of the lower number of clones we got. In the third patient, you see the general distribution is quite spread rather than a focus on some of the representative clones. So different individual does have a different representation, suggesting different individual must have respond differently to this infection. So from then to now, we have obtained several hundreds of human neutralizing antibodies, human antibodies against COVID-19 spike protein as well as RBDs. This slide highlights their binding to the RBD, to the trimer. I apologize for this kind of unclarity because of the tremendous amount of data included in this graph. But to take my words for the results, we categorize our antibodies for their binding activities. The higher the binder, the darker the color. So you can see among those clones, they have a tremendous difference in their binding activity to the COVID-19 spike protein, to the S1 protein, and to the RBDs, as well as some of ones have a cross-reactivity to SARS RBDs, and some even has a MERS RBD cross-reactivity. But I have to mention that those cross-reactivity antibodies largely confined to the antibodies targeting to the S2 domain. And then antibody recognized the NTDs and RBD in the up conformation or RBD in the down conformations are normally not cross-reactive. And then most of the neutralizing antibodies are confined to the regions recognizing RBD only. Although anti-NTD antibody also demonstrates some level of neutralizing activity, but far weaker compared to the antibody against RBD. And these results are very consistent with the results published in many groups in the US and in Europe. So it's quite consistent that the antibody against RBD are the major weapons to block viral infections in those individuals. And then once we have those antibodies, we developed this print assay very early on here in Shenzhen, because we have a laboratory 3 facilities here in collaborating with the local hospital, which allow us to quantitate their binding as well as neutralizing activity. So this graph highlights the raw data coming from the microscope. And you can see for 2 antibodies, normally we do a triplicate analysis. And then for each antibody, we do a 8 to 10-fold zero dilution. So you can see we quantify their neutralizing activity is quite unique and accurate, because many of the early on group try to use a CPE to quantify their neutralizing activity, which are quite difficult to judge because it's based on your eyes to measure. And then we convert those dots into the standard curve, a neutralization curve. On the left, we show their neutralizing activity against the pseudoviruses, and on the right is against live viruses. In overall, the 2 assays worked fairly well. And then, as you can see, different antibodies demonstrated different neutralizing activity. Those highlighted here, pointed by the green arrow, that the red, the purple, and whatever you call it, that color, that really represent the greatest neutralizing activity against the pseudoviruses and the live viruses. So this only the first batch of antibody we tested, and it was published very early. And then now we have much more antibody than early results, and demonstrated even more potent neutralizing activity than the early group of antibodies. So with those antibodies in hand, we really want to understand their structural basis for their strong neutralizing activity. So in collaborating with Professor Wang at the Institute of Life Sciences at Tsinghua University, we resolved both the crystal structure and also QuAR-YAM structures to understand their structure basis for neutralization. So I will show a couple of representative slides showing different antibody does have a different way of come to bind to the RBD, thereby to block ACE2 and RBD interactions. So you can see the first antibody on the far left is the 2F6 that we published already. You can see this antibody colored in purple and yellow bind to RBD from the upper, approach RBD from the upper left hand. And this binding, you can see this binding created a space disruption, the interference with RBD and ACE2 interactions here. And that will create a space clash. And that will push ACE2 to the sideways, thereby that disrupting the RBD and ACE2 interactions. The antibody in the middle, 1F11, bind to the RBD from the upper right corner. And this binding mode almost overlap completely with ACE2. So you could imagine that this binding basically block completely the antibody, the RBD binding, or the RBD and ACE2 binding. One of the control antibodies, CR32-2, I saw Dr. Boyd's presentation also include this one. It's really bind to the core domain of RBD, which doesn't interfere the ACE2 binding. That perhaps provides some structural explanation for why this antibody only bind but not neutralizing. But I have to say, recently some antibody has been identified to target this region, demonstrated some level of neutralizing. The mechanism of action of the antibody remain to be studied. So among those a few hundred antibodies, we picked 2 antibodies that move into the clinical development. And here on the right, showing the 2 antibodies that really bind to the different epitope on the RBD. And then we put these 2 antibodies into the ACE2 transgenic mice to see the protectivity of this 2 antibody combination in protecting animals from SARS-CoV-2 infection. You can see the controls of losing the body weight quickly, whereas the single antibody could attenuate that process, but a combination of antibody are performing the best among the oral group, suggesting the combination antibody does offer not just the potency, but also some other added advantages that presumably better than the single one antibodies. But the next slide really highlights the added value, not just to the potency, but also to minimize or limit the emergence of resistance, because we care about resistance a lot when you want to put into antibody into clinical use. So we have a fluorescent labeled VSV, a chimeric virus expressing the COVID-19 envelope in the lab. And then we use this virus to select the resistant strains against many different antibodies, including that classic clinical proven testing antibodies. So the 3 rows really highlighting in the increasing or decreasing concentration of antibodies, the likelihood of the viral growth. You can see when you plug the only single antibody into the culture, and then the green viruses come up regardless. Although the number of green are different, suggesting different antibodies has a different inhibitory activity. For instance, the first antibodies at the 250 nanogram per ml concentration, you could see have 19,000 green spots. The second antibody, on the other hand, have 35,000 green spots. But regardless, this within 4 days of in vitro culture, you're already able to generate resistant strain against this particular antibody. But if you put 2 antibodies together and then just everything else are identical, you can see the level of the number of the screens not only comes down, but also decreases. Only like 15,000 ones at a concentration of 1 microgram per ml, whereas a similar number was somewhere here, maybe at least 250-fold differences, suggesting combination of antibodies really not only improve potency, but also minimize the emergence of resistant strains. So these two antibodies are now, in fact, has already under the clinical trials. The phase 1 trial already started in July this year. We are already entering into the month 4 after the initial phase 1 trial. So this two antibody demonstrated a very good safety and the tolerability profile. And in the next week or so, we'll enter into a phase 2 or 3 trial here in China and in Hong Kong, and perhaps in other severely affected regions. While we are focused on those two antibodies, we also analyze the antibodies from different individuals and get their crystal structure resolved. So one thing we find very interestingly that Scott has already mentioned, the conversion or the share of public antibodies, that we identified among the first top 20 neutralizer or neutralizing antibody we have identified, we found a class of antibody that demonstrated exceedingly high neutralizing antibody. They all derived from the antibody heavy chains 3-53. So we went to crystallize them, and then we find they all approach RBD in a very similar angle. And then if you look at their epitope or their epitope on the RBD surface, you can see their epitope represent by the different color here. You can see here, so those antibodies come from 3 individuals, by the way. And their epitope are very similar. You can see they're all representing the patch, one patch here, one patch here. And then those epitopes are very similar to the ACE2 binding epitope, or the foothold RBD binding. And then combining their epitopes and also the angle they approach RBD. So it is very similar to the ACE2, the way the ACE2 binds to RBD. So we call them ACE2 mimicry, although not from the structure perspective, but also from the angle, not from the structure per se, but really the angle approaching to RBD and also the foothold, the footprints on the RBD. So those class of public antibody really, this is a great information for vaccine development, also for our understanding why majority of individuals are able to recover, hopefully, or at least contributed by some of those antibodies. So since they come from different individuals and able to generate the same potent neutralizing antibodies, it's providing some of the hope for the current vaccine development. And then we move further to see how those antibodies really bind to the trimeric spikes, to see why they have so much potent neutralizing activity. On the left, demonstrate the cryo-EM structure of this particular, this one member of this public antibodies. So you can see the blue and purple bind RBD only in the up conformation. And then since this is a trimeric structure, and then you can see all the trimer RBDs, all the RBD from this particular trimer will bind by this particular antibodies, 1F11. So you could imagine if you are a particle, this is a trimeric protein expressed on the surface of a particle, this particle will be covered by many of those antibodies, where thereby to block their interaction with ACE2. That perhaps explain part of the reasons why these public antibodies are so potent in neutralizing or in blocking viral interactions. The graph in the middle really highlights, I copied it from the German group, that's really highlighting the flexibility of this trimeric protein on the surface of the virus. So not just the RBD has its conformational changes, but also from the ester domain, it also has this hip, knee, and ankle that allow this trimeric protein swing around to find the best spot and angle to bind to ACE2. But unfortunately, the antibodies will block this RBD whenever it stands up, and then the trimeric, the 3 RBD will bind, all be blocked. So this antibody really is expected to block this protein's binding to the ACE2 form. Lastly, we also identify very interesting phenomena of this public antibodies feature. We find that in vitro, the graph shown on the right, in vitro, if you transfected trimeric S proteins into the cell surface, and then use those antibodies and several other antibodies to stain over time at room temperature, you find this public antibodies over time is able to trigger the S1 protein shedding from the surface of the transfected cell, whereas other 2 antibodies and control antibodies has no such effect. But if you mutate the S1, S2 regions and get rid of the positive charge residues mutated into GS-AS, and such a shedding effect will be minimized. And again, with other conditions, this wouldn't allow this shedding to happen, suggesting that this antibody is not only able to block the interactions between RBD and ACE2 when viral particle approaching the receptor or target cell, but even before that, this antibody can rip off the S1 proteins from the virulent surface and rendering the entire particle, at least some of the spikes, infectious, less infectious, or completely lost infectivity. So you could imagine at least additional mechanism of this antibodies neutralizing virus is to trigger a premature shedding of S1 proteins from the viral surface. And the shedded virulent protein, you could not only detect it decrease from this transfected cell surface, but also in the cell supernovae. So suggesting this is a really real phenomenon, whereas other antibodies and control antibodies doesn't have the same effect. So in summary, we are still in the process of characterizing more antibodies, understanding their individual character, and then also their correlation or causative relationship between diseases. But to summarize what we have learned so far, that from the beginning of the epidemic, we have already obtained several hundred of antibodies targeted to the SARS-CoV-19 trimeric spike and also RBDs. And then many of the antibodies demonstrate differential binding and neutralizing activity. We selected 2 most of potent and also complementary antibody for clinical development, and over the last 4 months of safety tolerability study demonstrated great safety profile. And in the next week or so, we will be able to get into efficacy trial in the 2 to 3 in human phase 2 or phase 3 study. So we have high expectation for that. And then with that, I have to thank the group that really working tirelessly since the beginning of the pandemic. Without their contribution, I wouldn't be here to present those results to you. First and foremost, the group that on the top left corner is my group from the Tsinghua University, group of dedicated graduate students working nonstop from the early years, including the Chinese Spring Festival. And then the second group is the Shenzhen #3 People's Hospital here. Again, passionate, dedicated, capable teams collect patients, running the facts, and then clone the genes, and then we still working on very, very well together. And Professor Wang on the right from the Institute of Life Sciences at Tsinghua University, providing the strong structural basis for understanding the antibody and SARS-CoV-2 interactions. last least but not least is the big clinical development team that putting those antibodies into clinical trial and hopefully will yield the best efficacy data in the near future. Thank you very much.
So thank you, Professor Zhang. So we have a question from the online attendees. So the question is that about the vaccine design, actually. So he asked, what is the target protein that is most appropriate for the SARS-CoV-2 vaccine design? It is the whole spike protein or some other segments of the like RBD. So this is a great question. I think I'm not at a position to say for sure, but I think both RBDs and spike protein should be tested. In fact, many of the vaccines being evaluated and will be approved soon are all spike proteins. But I know there's many RBD-based vaccines are being tested. So we are still in the early phase, understand in the full spectrum which antigen would be ideal for the long-term usage. So we'll wait and see. Thank you. So yeah, so any other questions that you can also type? So actually, I myself also have two very short questions. So the first one is that about the cross-reactivity, about the neutralizing antibodies you discovered. Have you tested any of your cross-reactivity with other coronavirus about your neutralizing antibodies? And what is your experience about the cross-reactivity of the current identified neutralizing antibodies, including yours and others?
So that's again very very important question. We have evaluated our antibody across about 10 different coronavirus spike proteins, RBD and STAs. Whenever there's a neutralizing, wherever there's a neutralizing assay available, we'll test their cross-neutralizing activity as well. So the conclusion is we have identified a bunch of antibodies that have really broad cross-reactivity against human coronaviruses and bat coronaviruses, but none of them are neutralizing. But we have identified some cross-neutralizer that are able to neutralize SARS-CoV-2 and SARS-CoV-1. Haven't identified any that are able to cross even beyond the MERS. So we are still early, but hopefully we will be able to identify the broad neutralizing antibody against all the pathogenic coronaviruses.
Thank you. So another question also about the neutralizing capabilities. So you mentioned you have identified at least 2 of them. They have very high neutralizing capabilities. So there are also others published about the neutralizing antibodies. So how is your neutralizing capability compared with the others?
They are all in a similar range of in vitro neutralizing activity, but we are uncertain how they perform in vivo because each one has recognized different epitope, different angles, different, and then we're in a different assay. So hopefully all of our antibody will demonstrate a good level of efficacy and safety in human.
Thank you. Thank you so much.
Thank you.